about class switching. So the first thing here, when we're talking about antibody structure, really you can organize this pretty simply into two regions. So you have the FC region, and the FC region, as you can see down here, this is going to be kind of the constant region. So that's what the C is. It's standing for fragment constant region. Okay, so the fragments are going to be all of these kind of little squares you see here. And sometimes these squares will get named. You might call this, region 1 and region 2 and region 3, but even when you get to that level of detail, you start to get beyond the step 1. So we just call these regions constant regions. And so again, what we highlighted here in orange is going to represent the heavy chain. Okay, so we have the heavy chain, and then we'll also have a light chain. Okay, and the light chain, you can see, I just remember the light chain, it's lighter. It's not as big as the heavy chain. So the light chain is this area that I'm kind of highlighting in teal here. Okay, so we have our heavy chain, we have our light chain, and then in the middle you can see that occasionally we'll have these disulfide bonds that are going to link these two portions of the antibody together. And so we already said the FC region is kind of like the constant region. Okay, this is kind of like the constant region. That's what that C is standing for. And then the AB here stands for antigen binding. Okay, so this is the antigen binding region, which actually makes a lot of sense. So if you understand some of these terms, you can kind of understand what's going on here. Let's just say, theoretically, we run into some bacteria. Okay, so I'm just going to draw some bacteria here. Okay, so here's some bacteria. And on this bacteria might have an antigen. And it might just so happen that this antigen binds to this portion of the antibody here in the FAB region. And so that's kind of where this name comes from, because the antigen binding will occur in this region. Okay, so the antigen might bind here. And again, there's going to be a very specific antigen that's going to bind to this FAB region, particularly the kind of distal end here of this region. And that's why this is sometimes also called the variable region. Okay, so let's recap some of this just to clean things up. So, there's a few different ways you can categorize this antibody. The first kind of way is we have a heavy chain, which is in the middle here. So this whole thing is your heavy chain. And then we also have a light chain, which is on the outside here. Okay, now the heavy chain you'll see is denoted with this H next to each of these letters here. So you can see there's an H, there's an H, there's an H, and there's different H's, right? This is going to be 1, this is H2, this is H3, right? So there's different ones, but all of these are representative of the heavy chain because it's in the middle. Remember, this is the heavy chain. That's why it gets the H. And then again, the light chain is denoted with the subletter L, right? Because that's the light chain. And then this letter here, there's a V and a C. So the C, again, stands for the constant region, okay? So the C is kind of the constant region, meaning it's going to be consistent, more or less, throughout similar forms of this antibody, which we'll get to. But there's also a region on the end here that has a V, and so the V stands for the variable region, okay? So the variable region is highlighted here in pink, and again, the variable region is going to be different for different antibodies, and these variable regions will have specific antigens that they can bind to, different shapes, different sizes, right, that these different antibodies can bind to. And the last thing that we talked about is you can split this up into a constant fragment, which is more or less going to be down in this region, and then an antigen binding segment, which is mostly going to be up here. Sometimes you'll see the term epitope used, and this is really just referring to the specific part of the antigen that variable region on the antibody is going to be binding to. So you might say that, this antibody is binding to its epitope, right, or the epitope on this antigen, that specific region that it can kind of match up geometrically to kind of fit into that space molecularly, okay? So that's what we're talking about when we say, that it's binding to its epitope. So the FC region, again, the FC region down here is basically going to consist of our heavy chains, right? So we have some of our heavy chains in the constant region, and this is going to determine the isotype of this antibody. In other words, when we say isotype, we're talking about things like IgM, IgA, IgE, right? All of these different immunoglobulins. So again, isotypes, you have your IgM, IgG, IgE, these are all different isotypes of the antibody. This region doesn't determine what epitope the antibody can bind to, right? That's for the antigen binding region. In the constant region, we're just determining the isotype. So what type of antibody is it? And in this region, we also have the C terminus. And so some people remember this by saying, hey, there's the constant region here, constant is C, and there's also a C terminus. And there's also, since we're talking about C, site of complement binding. IgM and IgG will use this region in particular for binding complement and then activating complement. So if we go back to the scenario where we have some bacteria here, right? So we just have some bacteria here, okay, that's floating around in the blood, and then potentially this bacteria might have an antigen on its surface, or a couple different types of antigens on its surface. Okay, and then what we might have happen is we might have, let's just say we have an antibody kind of floating around here, and it binds to one of those antigens. Okay, and again, it's going to bind using the Fab region, and then we have our constant region down here. Okay, so that's essentially what's happening. Now, once that binds, we can then activate the complement cascade, right? So we can activate the complement cascade, so we can get complement to activate its cascade and eventually lead to a membrane attack complex punching holes through that bacterial cell membrane. Okay, so that's what we're talking about with complement binding. And again, this happens with IgM and IgG. Specifically, the complement will get activated by this portion here, the CH2 constant region. So that is the portion where we will have complement binding. Okay, so I'm just going to write that in here. So complement will bind to that region on the FC portion like we shown here, and that's going to activate the complement cascade to then lead to the membrane attack complex coming in here and punching some holes in this bacteria. Now, the other route we can go here is opsonization, right? So it's kind of a similar concept. So again, if we take our antibody, so again, let's take our antibody binding here. And again, this is going to be the Fab portion. And then we have our FC fragment back here. And again, it's the same concept. If I have, some phagocytic cell around here, so let's just say that I have, let's just say that I have like a neutrophil or some phagocytic cell here. This cell can kind of get roped in to binding to this FC portion. Okay, so it's going to have a receptor here that can bind to this FC portion, and that FC portion will essentially stimulate this phagocytic cell to then undergo phagocytosis and digest this bacteria. Okay, again, binding to the FC portion, and so that's actually going to happen here at this third heavy fragment. This is where we're going to stimulate phagocytosis through opsonization. I'll just put that down here, opsonization. So the idea is if we can mark these bacteria with antibodies, we can have a few things happen. We can stimulate complement binding, we can stimulate opsonization, which will lead to phagocytosis of that bacteria. If we have enough antibodies around, we can essentially neutralize this bacteria or virus, right? We can bind to a lot of these antigen binding sites and prevent this bacteria from going on to reproduce or to infect different cells, whatever this pathogen is. By binding to multiple antigens here, we can essentially neutralize this pathogen. Okay, so the three things we can do: complement activation, opsonization, those are the big two, but we can also neutralize this bacteria by just basically flooding it with antibodies. So that's the tricky part. So in the FC region, we have a couple things going on. Complement activation, opsonization are going to happen at this heavy chain region. The variable region is what's binding the epitope. Remember, that's what's binding the epitope, binds antigens via the epitope, and this variable region will determine the idiotype. So the idiotype is referring to what this antibody can specifically bind to, what specific structure, specific epitope can it bind to, and with what strength can it bind to that epitope. So there's usually going to be a very unique antigen binding pocket, and that's referring to this idiotype. And this idiotype is going to be formed through things like VDJ recombination, which we'll talk about in the next slide. And just to put a plug in here, again, going back to opsonization, remember, what are the two big things that are going to kind of lead to opsonization? So this is particularly high yield. Remember, it's going to be IgG and C3b. Okay, so when you're thinking opsonization, right, when you're thinking phagocytosis, think of the constant heavy region 3, right, this region closest to the C-terminus on IgG, and then also keep in mind that complement is going to be, again, like we said, at the CH2 region here. Okay, so now that we've talked about some of the structure of antibodies, let's focus in on some of the properties of antibodies. And really, we're just focused here on the high yield information. So looking back at our antibody structure here, we can see, again, we have our variable region that's going to be up here in the pink color. And then we also have this constant region that's down here in the blue color, as we just talked about. And this is functionally how we want to kind of think about dividing the structure. And again, this variable region, it's going to contain the area that we'll call the idiotype. Okay, so the idiotype is going to refer specifically to the binding of this antibody receptor to some epitope, right? So that's some region on our antigen. So the idiotype being this structure here. And again, the constant region representing the area where we have our isotype. The isotype representing what type of antibody do we have? Is it going to be an IgM antibody, an IgG antibody, etc.? Okay, so we have the idiotype up here, the isotype down here, again, the FC region, and then we have the FAB, the antigen binding region up here. Now remember that this can also be divided based on the types of chains that we have. So we have a heavy chain, right? So the heavy chain is going to be this chain right here down in the middle. And then we also have a light chain, okay? So the light chain is going to be this one that's on the outside, only in the antigen antigen binding region, the FAB region, right? So that's how we separate this. And again, the thing that you want to remember is that there's different genes when we're talking about our gene transcripts that kind of get shuffled around randomly to create a whole bunch of different idiotypes so that we can bind to all types of antigens, okay? And those shuffling of genes is sometimes referring to specific segments here, VDJ recombination. And you can see that this D is in parentheses. The reason for that is because the heavy chain segments use the VD and J shuffling, whereas the light chain segments primarily are going to use the V and J shuffling. So this is just a little thing just to be familiar with. Again, it's these little details that sometimes come up. But the thing that I want you to kind of take away from this is that this whole process of VDJ recombination, if you think about it, right, you have some B cell, and inside of this B cell is going to have these genes with the V segment, with the D segment, with the J segment. It's going to have those genes inside of the nucleus, and it's going to shuffle those genes around until we get some unique immunoglobulin receptor, right? So we shuffle all of these genes around, and then we come out with this very unique idiotype on this B cell receptor. Remember, what is a B cell receptor? A B cell receptor is essentially a membrane-bound antibody, right? So it's like we created an antibody, like we see here, we just stuck it on the surface of a B cell. Specifically, IgG and IgM are going to be kind of notorious for being on these membrane-bound surfaces for B cells. You can also see this with IgD a little bit, as we'll talk about. But the point is this VDJ recombination is going to happen in these B cells, but there was no antigen that stimulated this process. This process happened as B cells matured. When B cells were maturing, where are they maturing, by the way? They're maturing in the bone marrow, right? We talked about that. While these B cells are maturing in the bone marrow, they're shuffling these genes around, these V, D, and J genes in the heavy chain and the V and J genes in the light chain, and they're making unique idiotypes, right, that are going to go on the variable region in the fragment antibody portion that we just talked about right up here. Okay, so that's what this whole process is. Again, the big thing I want you to remember is that this is going to be antigen independent. Okay, so there was no antigen that stimulated this process. It's just going to happen as B cells mature. We're going to make a whole bunch of different receptors, right? That's the point. And that's why this is completely random. It's random recombination of these genes. But you got to remember, we're doing this amongst millions and billions and etc. B cells, right? On a mass scale. So we're going to get all different kinds of receptors. And ideally, certain receptors are going to favor the binding of future infections, right? So that's the concept. And this is all going to happen in the fragment antigen binding portion up here, which is where we're going to bind those cells, right? The constant region, we're not binding antigens in the constant region, right? The constant region we talked about in the last video, it's about complement binding, it's about opsonization, phagocytosis, etc. So that's the concept here. Now, when we go into this next category here, somatic hypermutation, affinity maturation, isotype switching. So these are things that we've also kind of already hit on a little bit if you've been following the series so far. And really, somatic hypermutation, essentially think of this as, once we've interacted with that antigen, so we go back to our B cell here, once we've actually kind of found our antigen, so let's just say that we have some, we'll draw some pathogen here, maybe it's a bacteria, right? So we have some pathogen here. And again, on the surface of this pathogen, we might have an antigen. So let's just say that there's some antigen here, okay, some antigen here. And then our B cell receptor might interact with that antigen. It might be the perfect fit, right? We might find our epitope here on this antigen that this B cell binds to, and it's a perfect match. And then what happens, right? These B cells can potentially get activated. Remember that B cell can then kind of internalize some of that antigen potentially, and then redisplay that antigen in MHC2 to then activate T cells if this was a T helper dependent response. If it was T helper independent, this B cell might just turn into a plasma cell potentially and release IgM. Okay, so these are all things that we talked about in the B and the T cell video. But again, this process, once we activate this B cell, these B cells, some of these activated B cells can go to lymph nodes, right? And they can go into those secondary follicles we talked about initially, those pale staining germinal centers, and they can proliferate. And so you get a whole bunch of B cells proliferating in the lymph node, in those germinal centers. And as they're proliferating, what these B cells are doing is they're essentially saying, look, I know this receptor binds to this antigen, but what if I just tweak it a little bit, right? Maybe I'll just add maybe an extra couple peptides here or amino acids here. Then let's see if it binds more tightly or more weakly. And essentially the B cells are doing this in the lymph node on a very large scale. So they're randomly mutating the known binding receptor and trying to see if we can get it to bind even stronger. And those receptors that do bind the strongest survive. And that's called affinity maturation. We're essentially maturing the affinity of the receptor. We're finding the receptor that binds the tightest. And how do we do that? Well, we do random variations, again, in the variable region. Same region, right? We're just making little tweaks to the variable region that we already know binds to the antigen. Now, that's different from VDJ recombination. VDJ recombination, we're just mixing up stuff. We don't know what we're doing. We're just shuffling genes everywhere, and we're just trying to make a diverse pool of B cells. With affinity maturation, we're looking for something that is already bound to its antigen. We just want to make that binding stronger. And for that reason, this is going to be antigen dependent. Okay, so this is going to depend on the antigen that binds to it. That's what's going to trigger this whole process. So somatic hypermutation, affinity maturation, here, let's kind of highlight these. These are going to be antigen dependent. Okay, isotype switching is as well, which I'll talk about in a minute. And then VDJ recombination, that's antigen independent. Now, why is isotype switching antigen dependent? Well, you can kind of put the pieces together based on what we've already talked about. So remember, the whole point of isotype switching is after this B cell gets activated, what is it going to do? Well, like we just said, it's going to internalize this antigen, right? So it's going to take that antigen, it's going to internalize it potentially, and then after it internalizes this antigen, it can display that antigen on MHC2 receptors, because remember, B cells are antigen presenting cells. So let's say that there's some MHC2 receptor here, okay, so the B cell can then display it, and this is really the same song and dance that we keep talking about. So this B cell then is just going to go to a T cell. What's that MHC2 receptor going to bind to? It's going to bind to a T cell receptor, CD4 co-receptor, right? 4 times 2 is 8, rule of 8s. So that B cell, that's an antigen presenting cell, then presents this to a T cell, then it becomes activated. And that T cell will also have this CD40 ligand that binds to CD40 on the B cell. Remember, this is particularly high yield, and we've talked about this a couple times. So that binding between the B and T cell is going to stimulate class switching. Okay, so we have kind of two receptors binding, MHC2 binding to the T cell receptor and the CD4 co-receptor, and then we have CD40 ligand binding also, like we talked about before. And if this is unfamiliar, go back, check out those previous videos on B and T cells. We'll go into this in greater detail, but essentially that binding is going to stimulate class switching, and then we're going to have certain cytokines that are released that'll stimulate different versions of class switching. If we want to make IgE antibodies, right, we might see interleukin 4 and interleukin 13 being released. If we want to make IgA antibodies, we might see interleukin 5. So again, you want to remember these specific cytokines as they're particularly high yield to note in this isotype switching. But going back to my original point, how come this is antigen dependent? Well, it took an antigen to start with to get this whole process going, right? We had to internalize this antigen, display it on MHC2 to eventually present it to a T cell to get isotype switching. So that's why both of these are antigen dependent. Again, VDJ recombination, antigen independent. Now, the big difference between these two is that somatic hypermutation, affinity maturation, we already said it's taking place at the variable region, right? It's taking place at the variable region. We're slightly changing this idiotype so that it gets the strongest binding to our antigen. Whereas with isotype switching, what we're doing is we're changing the constant region. So if I change the constant region, what can I change? I can change it from IgM to IgG to IgA to IgE by using these cytokines, right? So that makes sense that we'd be changing the constant region in isotype switching. Now, the last thing I want to talk about here is the primary response versus the secondary response. And this is actually pretty intuitive to kind of understand. So let's just say that this is time, and then let's just say that this is antibody concentration in the serum. Okay, I'll just put an S there for serum. So this is my antibody concentration in the blood or the serum, and then I have time here on the x-axis. I'm going to move it over so we can see it better. Okay, so this is time, and then we have antibody concentration in the serum. Now, let's just say that at some point here, we are infected with some antigen. Okay, so we'll just put a 1 here to signify that this is the first time that we've seen this antigen. Maybe it's a bacterial infection. Okay, so let's just say that we have some bacterial infection. This is the first time we're seeing this antigen. So what will happen is initially we might have some stable level of IgM that spikes. Okay, so our IgM level, after realizing that we're infected with this antigen, again, we're going to have an inflammatory response, and some B cells will recognize this antigen for the first time and turn into plasma cells and increase the amount of IgM being released. Okay, so we'll get some IgM release here. So we might also get some IgG being released. And the IgG response we'll get after class switching is actually going to be much greater than that initial IgM response. Okay, it's going to take B cells a little bit of time to undergo affinity maturation, isotype switching, but eventually we're going to generate generate an even stronger response using the IgG, all from that first exposure. And now what will happen is, remember, after we have an initial exposure, there'll be memory B cells that are around for future events with this antigen. And let's say we get infected with that same antigen bacteria a second time. Okay, so the second time, what's going to happen to the IgM concentration? The IgM concentration is going to do pretty much the exactly same thing. Okay, it's not really going to change much, right? The peaks here are about the same. Okay, and the amount of time it takes for IgM to respond, it's pretty much all going to be same song and dance. IgM doesn't involve class switching. It's kind of like your standard antibody, right? Even the T helper independent responses will still release IgM, like we talked about in the B cell video. However, the difference here is that now we have some of those memory B cells that are around, and they've already undergone class switching, and they have very strong affinity maturation and high levels of IgG that can be produced. So what will happen is we'll get a slightly faster and much stronger response from IgG. So I'm just drawing a massive jump here in your IgG response. So we have an exponential increase in our IgG response this time. And that's because we have memory B cells that have already undergone this whole process, really, right, from some kind of T helper dependent response that are now generating, significantly more amounts of IgG. And specifically, that IgG is going to come mostly from plasma cells, right, from this memory response, okay? So again, the key is on that second exposure, you're not really seeing a huge change in IgM in terms of timing or the absolute concentration of it in the blood serum. However, on re-exposure, you can see there's a lot more IgG that can respond actually slightly faster even potentially. And this is where you see things like, if you want to see if this person was ever infected with a certain bacteria, you can try and draw their antibody to that bacteria. Or if you vaccinated somebody, you can see their titers, right? You can see how much IgG, how much of a response they generated when they were vaccinated. And so you can see if they're immunized by having a set level of IgG after re-exposure. All right, let's wrap up antibodies by talking about immunoglobulin isotypes. And here, the important thing to know is really the structure of some of these isotypes, and then when do we specifically see them in terms of their function. So let's start off with IgM, which is kind of like our base immunoglobulin, right? Because with IgM, if we have a T helper independent response, or this is a first response, usually it's IgM that we're gonna see initially. Now, IgM can be a pentamer or a monomer. Now, classically, it's thought of mostly as a pentamer, and you can see an example of that down here. So you can see this IgM molecule, and you can see that there's all of these different binding sites here on this IgM molecule. So you can think of these as like individual kind of antibodies based on the antibody structure that we talked about before, and you can see that they're all linked to this structure in the middle here, and this link is the J chain. Okay, so that's going to be this linkage kind of in here, call that the J chain. Okay, so they're linked by this J chain or J protein, and again, if you look at how many antibody binding sites there are on one of these pentamers, you can see that there's 2 for each antibody, which is pretty much the case across the board. Every one of these antibodies is going to have 2 antigen binding sites, which means that a pentamer of IgM is going to have 10 binding sites, right? So it's going to have 10 binding sites for antigens. Now, the reason that's significant, if you think about some of your antibodies that are monomers, right? So, let's just say that you had an IgG antibody that already underwent affinity maturation for some antigen, and it's really strong binding, right? We've already had a response to generate this IgG antibody. And so we can say that this IgG antibody in particular might have a higher or stronger affinity for an antigen. Maybe it already had a T helper dependent response where we had a B cell that went to the lymph node and then underwent affinity maturation. And that B cell is now a plasma cell producing IgG to our antigen. And now we can say, okay, well, maybe this IgG antigen then has a higher affinity than that same IgM antibody, right? An IgM antibody with the same idiotype as this IgG might have a lower affinity. Okay, and this might vary in real life, but again, just for comparison's sake, let's just say that for those reasons, we have a higher affinity IgG and a lower affinity IgM. Where this gets tricky, where this comes in questions, you have to remember this IgM immunoglobulin has 10 binding sites for antigens. So it's got 10 binding sites for antigens. Now, if you compare that to IgG, IgE, IgD, this only has 2, right? It only has 2 antigen binding sites. So that's kind of tricky, right? So one has 2 and one has 10. So what we call this, we could say, look, even though this IgM immunoglobulin might individually, each one of these sites might have a lower affinity, right? So each one of these sites, so each one of these idiotypes here might have a lower affinity. Overall, the whole molecule, if we add up all of the binding from all of these individual sites, right, we might actually have an overall increase in its ability to bind antigens. And so that we would call the avidity. Okay, so that's if we totaled up all of these different sites together, right, that would be the avidity. Avidity, right? And so the affinity is we're just referring to that one site. And so we might say that this IgM antibody potentially might actually have a higher avidity, right? Because it has so many more binding sites as opposed to the IgG. Now, again, like I said, there's specific examples of this that might be different, but this is kind of a high-level way to just think of the terminology when they use avidity versus affinity. Okay, just cleaning things up here again, let's just recap some of this. So again, IgM, usually referring to the pentameric structure. Okay, we said 10 binding sites linked by a J chain. Early response to infection, that's what we just talked about previously in the last video, where we initially, that primary response is going to come out IgM initially. And again, when we have repeated exposures, we have a similar response on repeated exposures to the antibody serum concentration of IgM. We talked about that in the last video, okay? So go back and check that out. In terms of its function here, the primary functions of IgM are going to be to activate complement, and that's part of the reason for that is because it's so big. But remember, it also acts as a B cell receptor. So also IgG is kind of the other big B cell receptor to be familiar with. And I feel like we've kind of talked about a lot of this already. Let's transition over to IgG now. So IgG is the most abundant immunoglobulin in the blood that's particularly high yield. Also remember, this is now a monomer. Okay, so we're talking about monomeric structure generally versus the pentameric structure typically of IgM. And again, this is going to be a little bit more of a delay in terms of its response to an infection when we compare it to IgM. Usually you're going to get your IgM being released first. However, if there's repeated exposure, right, you're going to get greater responses of IgG because a lot of that is going to come from plasma cells that have already undergone class switching. And so those plasma cells and memory B cells are going to be stimulated to have a greater IgG response. And that's why we use IgG for things like titers, like we talked about in the last video. And again, the big functions of IgG, also activating complement, we already talked about that. That's primarily going to be done in the FC portion back here, and opsonization also done in the FC portion. Remember, opsonization, there's two really big opsonins that you want to be familiar with for step one. IgG is going to be one of them, and the other big one is C3b, like we talked about a little bit earlier in the series. Okay, so complement C3b is one of the classic opsonins, and IgG also will do this. And the way that we're doing this is if we have some antigen that we're binding to, right, on the surface of a bacteria. So we're just going to draw a big bacteria here. Okay, so let's just say that, our immunoglobulin is bound to this bacteria. It's going to have a region there in the FC portion where essentially you can kind of pull in a neutrophil here. So maybe there's some neutrophil here, okay? And this region can essentially kind of lasso in the neutrophil to bind here and then phagocytose this bacteria. Okay, so that's what opsonization is. That's what we're talking about. And again, largely driven by IgG and C3b. Also, the IgG structure, like the IgM, both of them can act as B cell receptors. And we'll talk a little bit more about this coming up when we talk about things like hemolytic disease of the newborn. But the big thing you want to remember is that the IgG antibody, because look at how small it is, right? In general, it's a lot smaller than the IgM. And so for that reason, that's how you kind of remember that the IgG antibody can be transferred through the placenta from mom to the fetus. Okay, so what mom's doing is mom already has antibody. Throughout mom's life, she may have been vaccinated, right? She may have had multiple types of infectious agents that now she has a memory response to, and she has those IgG immunoglobulins floating around in the blood, and those can go to baby until those IgG immunoglobulins are turned over. Okay, once baby is delivered, mom's not going to be supplying IgG immunoglobulins through the placenta anymore. Okay, so initially that newborn might have some amount of mom's immunoglobulins that are eventually going to start to kind of come down during the first few months of life. And so how does that newborn continue to get some immunoglobulins from mom? Well, that would be through IgA, through breast milk. After the baby is delivered, right, through the breast milk, we can give IgA antibodies. And that's how you remember that IgA antibodies are associated with things like mucosa, mucosa-associated lymphoid tissue like Peyer's patches, or the mucosa of the GI tract. So again, classically in breast milk, also sweat and saliva, and IgA initially in circulation, it's more of a monomeric structure like this, but again, it's linked by a J chain like we saw with the IgM. Here's the J chain again to form a dimer once it's into the mucosa. These IgA antibodies, they don't activate complement. And one big pathology you'll see IgA come up a lot in is going to be, and there's a few of them, but this is one of the big ones, is celiac disease. Okay, and the thing that sometimes gets asked about in celiac disease, the classic antibody that you want to be familiar with is going to be tissue transglutaminase. Okay, so tissue transglutaminase, and sometimes they'll just write it as TTG. Okay, so tissue transglutaminase, and they'll sometimes ask you, you'll be like, oh yeah, I know this is for celiac disease, but they'll ask you which antibody specifically, which isotype is it? And it's IgA, right? Celiac disease is primarily affecting small bowel in general and absorption. And again, when you're thinking small bowel, when you're thinking mucosa, GI tract, you're thinking more IgA, right? So this is an IgA antibody. All right, let's jump down to the IgE antibody. So the IgE antibody, also a monomer like IgG. And the thing that you want to remember with IgE, there's two big things. It's really going to be its ability to respond to helminthic infections. So we're talking about worms, parasites. Also, its effects when we're talking about type 1 hypersensitivity reactions and things like asthma. We have a whole video on that already, so I don't want to go into too much detail in this video, but the concept essentially is, remember, you have mast cells and basophils that are housing essentially a lot of the histamine, proteases, leukotrienes. So let's just say that I have some, I have some mast cell here. Okay, so we're just going to call this a mast cell. Okay, so here's my mast cell, and this mast cell will have on its surface a specific type of receptor. We're just going to call it Fc epsilon 1. Okay, and it's not super important to know that, the exact name of all of these, but the point is that this receptor, like it says, it will bind to the Fc portion of antibodies. And so what do we mean by that? So hold that thought on the mast cell and let me show you. So and let's say that we have some patient that had been exposed to ragweed pollen or bee sting or something like that, and initially the initial response, they probably initially responded to IgM. Okay, so initially we might have had some B cells with IgM receptors, for example, and let's just say that this antigen bound to the IgM portion of this immunoglobulin. And what will happen is those B cells that have IgM on their surface, for example, that bound to this specific antigen might go and present this to a T cell. And they might undergo class switching. And so then what happens? And so then we start to make plasma cells that start releasing IgE antibodies through the stimulation of interleukin 4, like we talked about before, and interleukin 13. Okay, so now if our patient comes into contact with ragweed pollen again, or if they get a bee sting again, they have some plasma cells that are releasing IgE to our antigen. Now what can happen is some of these IgE antibodies can actually make their way and bind to the surface of mast cells. And look at how it's bound, right? So here's the Fc portion bound to the Fc receptor. And remember, this is your Fab portion out here. Okay, and now if we bind, remember we have the same idiotype as this IgM receptor because we went through class switching. And now let's say that we have a second exposure to the ragweed pollen, for example. So now, right, we're going to bind to that same region, okay, because we have the same region, we have the same idiotype. But now what happens is with these IgE antibodies on this repeated exposure, they're all bound to these mast cells. We might have a whole bunch of them here. And they can cross-link once they bind to their antigen. And that will stimulate these mast cells to degranulate. And so these mast cells, because of this binding, right, of the IgE receptor on its surface, now bound to its antigen and cross-linking, these will start to release substances, histamine being a big one, and leukotrienes, okay? So all of these will start to get released, and we call this process degranulation, okay? So the granules that are housing all of these structures are releasing them. So we call this degranulation. Okay, so the mast cells will degranulate, release all these substances, and you can imagine if you have repeated exposures, right, you're going to make more and more of these IgE antibodies, and more of them are going to be sitting on these mast cells. So that's why if you're allergic to something and you get exposed to it multiple times, even if it's certain medications, can actually go down this route of anaphylaxis, because once you get to a significant enough amount of IgE antibodies bound to the surface of these mast cells and basophils, then what will happen is when you get exposed to that antigen, you're going to have so much degranulation that you can actually push this patient into anaphylaxis, okay? And so that would be the concern. This is a type 1 hypersensitivity reaction. We're hypersensitive to some antigen from repeated exposure. Now, in general, like we said, the IgE response is going to be particularly effective against helminths, and we can remember IgE with eosinophils, okay? And lastly on here, we have the IgD antibody, which is not particularly high yield in general. We won't go into too much detail on this. Just remember that it's a monomer. So the big things here, like we said, just make sure you're familiar with the different structures of the immunoglobulins, their function, and then any specific pathologies they're involved with. For example, IgE, we talked about type 1 hypersensitivity. IgA, we talked about celiac celiac disease in breast milk, IgG crosses the placenta, classic and opsonization, IgM acting in the early initial response, and then it's a pentamer. So just remember the key facts here. A lot of what's on here is a lot of what you have to know to distinguish some of these isotypes. Take 15 seconds to pause the video and read the question, and then we will go through the answer. You know, there's always certain questions that really kind of, either get under your skin or get you a little nervous. And this is definitely, for me, one of those questions. You know, just looking at this, it looks deceivingly short in terms of the question stem. But you see this table, you see some kind of complicated data, and it can be a little bit intimidating. So, let's just kind of walk through this here step by step. So, a researcher conducts an experiment to measure the effects of thymic tissue in adult mice. And again, you might not necessarily think of this the first time reading this, but again, thymic tissue in adult mice, right? When you're thinking about thymic tissue, at least, when you think about adults, you think about thymic tissue more so in newborns, right? Interesting that they're talking about adults here. So a control group and 2 additional groups of mice are studied. All right, this is important. We have to kind of stay organized in questions like this. So we have a control group. Okay, so we have this control group, and then we have 2 additional groups here. So it looks like group 1 mice undergo placebo surgical intervention, omitting thymectomy. Okay, so it looks like it's a surgical intervention essentially, but they're not taking out the thymus. Okay, so that's group 1. Okay, and then we have a second group here. So group 2 mice undergo surgical thymectomy. Okay, so group 2 go under for thymectomy. XX77, a primary immunization, is given 11 days prior to surgical intervention. Okay, we're not sure if this is relevant yet. And a booster immunization is subsequently given 21 days later. All right, and we can see that referenced here a couple times. All right, what's the question here? So the data below is collected at 30 days after surgical intervention. Okay, which of the following can the researcher most reasonably assume based on the results of the experiment? Okay, so again, kind of appears like a tough one. And, these things where you have to kind of keep track of the days here, right? So the primary immunization is given 11 days prior to a surgical procedure. So let's just write this out here. So here's the surgery, right? So they gave the primary immunization here. This would be let's just say this is day 1. Let's just say that this is day 11. And I'm assuming this booster is given 21 days after the primary here. So we can just say day 21. Okay, so just to kind of give us an idea. Now, the data below is collected at 30 days. Okay, so the data is collected at day 30. So this is where the data, just write data, is collected from. Okay, so this is the data in this table here. CD19, CD21 positive cells. So again, you got to know some of these markers, right? So these ones we've talked about in the series so far. So these markers are going to be consistent with B cells. Okay, so CD19, CD20, CD21, usually referring to B cells, and you should know that. I noticed that the control group has slightly more in the way of overall B cells. So the control group, I'll just put control, has a little bit more B cells, right? Group 2, slightly more than group 1, okay? If we look at the second column here, there's a pretty significant difference in the percentages here. So now this is XX77, this is the vaccination, specific heavy chain variable region mutation frequency. You read something like that, and that can definitely start to give you some palpitations on test days. So we're not going to panic, we're going to break this down. So heavy chain variable region, okay, so remember your immunoglobulin, right? So your immunoglobulin, you have your heavy chain, right? So you have the heavy chain, and then you also have your light chain, right? So you have your heavy chain and you have your light chain, and it's talking specifically about the variable region. So this region out here. Now remember, what's the significance about the variable region? The variable region, right, is going to be the idiotype. In other words, this is where we're going to have antigen binding. And we're going to have a lot of different variability in this, right, from birth with things like VDJ recombination, which we can see here is one of the options, right? So anyway, this XX77 specific heavy chain variable region mutation frequency. So showing how many different mutations there is in this region that can bind to this antigen essentially, right? So we can see in the control group, we have quite a bit more in terms of the percentage of mutation frequencies. If we look at group 1 and group 2, we can see that these percentages are a bit lower than what we see here with the control group. Right? What does this mean? Well, it's kind of telling us that, look, this variable region, right, when we talk about things like somatic hypermutation or affinity maturation, remember what this basically is, right? If I give somebody a vaccine, or somebody's infected with a pathogen potentially, initially, right, my B cell, let's just write in CD19 positive, CD21 positive, right? Because that's what one way to kind of make this stick, right? So this is our B cell. And so this B cell, initially, when it presents, right, it's going to phagocytose some pathogen. It's going to present a protein antigen, right? So it presents this protein antigen. And let's say it presents this to, let's say this is a CD4 positive T cell. So essentially what happens is this T helper cell can do a couple things, right? We already said one thing that can happen is this CD4 positive T cell can cause this B cell through CD40 ligand, can cause this to undergo class switching, right? Class switching, which this would change not the idiotype of the antibody, but this would change the isotype. That's going to change the heavy chain here, right, in the constant region, so that we can make things like IgG, IgA, or IgE, right? So that's one thing that can happen. The other thing that can happen is this CD19, CD20, CD21 positive B cell can go to the lymph node, right? It can go to the lymph node. It can head to the secondary follicles, right? We talked about these. And these secondary follicles, particularly in the germinal centers, kind of in the middle where you have more of the pale staining, these germinal centers are areas where we have a lot of these different B cells, right, that have this specific antigen binding, much like we see this XX77 specific antigen. And what these B cells are doing in the germinal centers here, in the secondary follicles, is they're essentially basically taking this receptor right here, right? They're taking that very specific idiotype. Because remember, what is a B cell receptor? A B cell receptor is essentially going to be an immunoglobulin, right? Very similar to an antibody. And so what we're doing here is we're taking that specific receptor, right? So maybe this is my heavy chain, here's my light chain, and we're just making, fine-tune adjustments to it. We're just doing a bunch of random mutations. Maybe one B cell has a mutation here, and maybe another, B cell in this secondary follicle has a mutation here. And they're all just kind of making little mutations, and then they're going to see out of all of these B cells which one's mutations have the strongest binding. And so the idea is if we have more mutations happening in that very specific site, in the heavy chain variable region of this specific antigen binding site, then we're going to have a higher likelihood of stronger binding, right, for this B cell. Okay, so this is why, and we'll get to the other options in a second, but this is why the answer is impaired somatic hypermutation after surgery. Okay, the key other point to this is after surgery. Now, if the options were, after thymectomy and after surgery, and you're like, oh, I don't know which one to pick, the answer is after surgery, right? Because in group 1 and group 2, in both situations, even though group 1 still has a thymus, right? Even though group 1 still has a thymus, the lower level of somatic mutation is still there. In fact, it's actually even lower than those mice that underwent thymectomy because they have a lower amount of these mutations in that idiotype, in that specific heavy chain variable region. Let's just go through the rest of this here really quickly. So XX77 specific IgM. Okay, so now we're talking about immunoglobulins that are IgM that bind to this antigen. And we can see that the levels of these, right? So this is just like a quantitative level here. So, the control, the group 1, group 2, they're all pretty consistent. I mean, nothing is like even, 5% different really, I don't think. So they're all pretty similar. The IgG levels, kind of the same thing. I mean, very small variation here. Now, what does this tell you? Well, remember if we go back to what I said earlier, right? There's two kind of pathways here, right? There's two pathways here. I'm gonna highlight them in red. The first one, right? You can go through class switching and you can change the isotype. The other thing is you can do somatic hypermutation to these B cell receptors, right? To these immunoglobulins, and these can go on to change the idiotype, right? So the idea here is we already know that the level of mutations in the idiotype are lower, right? Which is consistent with somatic hypermutation, impaired somatic hypermutation. But we can see that we're able to get some class switching, right? Pretty consistent with the control group who, didn't have the surgery presumably. So in all these situations, these are all pretty similar. So we can assume from this then, right, based on this data, that it's unlikely that there's impaired isotype switching after thymectomy, right? So if you just look at thymectomy, which is group 2 here, right? Group 2 actually has more IgG than group 1 or the control group, right? So impaired isotype switching is pretty unlikely. The same would be true for impaired CD40 ligand binding, because remember, we need this to get class switching, right? If we don't have CD40 ligand binding, it's going to be pretty hard to get class switching. That's why if you have a defect in CD40 or CD40 ligand, we call it hyper IgM syndrome, because then all you can really produce is your baseline IgM, which is not what we see here, right? So in both of these groups, we can clearly see that there is an IgG response. Now, impaired VDJ recombination after thymectomy. Now again, VDJ recombination is going to usually happen much earlier on. We're talking about
adult mice here, right? And so it seems pretty unlikely, right? And if we're talking about adult mice, that this would be the most appropriate answer, because again, this is happening a lot earlier on, and we really don't have anything here to suggest that we have impaired VDJ recombination. There's clearly a better answer with somatic hypermutation. Finally, the last option here, marginal zone B cell hyperplasia. So remember, if you're talking about B cell hyperplasia, marginal zone, particularly in the lymph node, right after surgery, well, let's take a look at this. So here's the B cells. Now, hyperplasia would suggest more B cells, right, in this marginal zone. Now, if I look at, let's see, after surgery, so we're saying that essentially group 1 and group 2 have hyperplasia. If I look at group 1 and I look at group 2 in terms of the levels of their B cells, remember, they have less than the control. The control has more B cells than group 1 or group 2. So it doesn't really make sense that the marginal zone B cell hyperplasia would occur after surgery, right? Because if it did, I would actually expect to see more B cells in group 1 and group 2, which is not the case here. The control has more B cells. So for those reasons, impaired somatic hypermutation is the best answer. Things to take away from this. If you know your immuno, if you've been following the video series, you're building on the concepts, a lot of what's talked about here, you're just like, oh yeah, I've seen this like 5 times. You've talked about this so many times. And that's a good thing, right? Because if you're at that point and you've watched all these videos and you're like, yeah, I really get this now, that's fine. If it's annoying to hear the same thing over and over again, that's a good thing. However, if you're kind of going through these questions and you kind of back off a little bit when you start to see some of the details here, just go back to your basics, right? Even if you've been watching these videos all along and you get to this question and it's a tough question for you, go back to your basics, right? What do you know? What can you rule in? What can you rule out? And how can you try to break down some of this data with the topics that you feel the strongest on so far in the unit, right? And that's usually the best way to go about it. So hopefully that helps.
Hey everybody, Dr. Viso here. In this video, we're going to talk about complement, and we'll talk a little bit about the general properties of complement, some of the functional components to be aware of. Some of them we've kind of already touched on in the previous videos, and then we'll go through the 3 major pathways, all leading into the common pathway, and then we'll talk a little bit about some of the inhibitors, which makes a really nice transition into some of the disorders of complement, which we'll also touch on. So just to give a very high level overview here, when we're talking about complement, we're talking about plasma proteins that are typically going to be synthesized in the liver. When they're synthesized, again, they're in an inactive state. So they might be in the serum, but that doesn't necessarily mean that they're going to be setting off complement cascades on pathogens. Okay, so usually there's something that is going to trigger activation of the complement system. Okay, so once we get activation of the complement system, which could be by immunoglobulins, it could be by enzymes, certain carbohydrate moieties, which we'll discuss in the next video. Once we get that activation, there's 2 kind of endpoints to this complement cascade that you want to be familiar with. So the first one is going to be C3b. Okay, remember, so C3b is going to be very much involved with opsonization, and we talked about that previously. C3b and IgG being the 2 particularly high yield things to be familiar with when you're thinking about opsonization, when you're thinking about triggering that component of phagocytosis. Now, the other kind of endpoint to complement is going to be the membrane attack complex, which that's what this MAC stands for, this membrane attack complex. And as we go through complement, we're going to kind of start at the top. So, we'll start with C1. Okay, so we're going to start with a C1, but then eventually, as we go through the complement cascade, we'll eventually end with this C5, C9 complex. Okay, and this is sometimes referred to as the membrane attack complex, or the MAC. Now, that doesn't necessarily mean that we go, exactly in order here. Okay, so that's not true, but we do start at C1 and then we end at C5, C9. Okay, and then everything in between is what we'll discuss in the next video. Now, do you have to understand every single component of complement? Not necessarily, right? It's not particularly high yield to know that, but there are certain functional components that are very high yield that you want to be familiar with. And a lot of these we've kind of already touched on, so this should be a little bit of a review if you've been following the video series. So we have C3a and C4a, which, remember, sometimes you'll see this written as anaphylaxis, right? So they're going to stimulate anaphylaxis. Now, we're not talking about anaphylactic shock necessarily, but the term anaphylaxis is simply referring to histamine release from mast cells, which is pretty consistent with what a patient experiences when they go into anaphylaxis, right? And so C3a and C4a will be promoting that process that ultimately leads to vasodilation. Now, a lot of this is really just kind of promoting the inflammatory cascade that we talked about in the acute inflammation video. Now, remember, C5a is very high yield because C5a actually does, the same thing as C3a and C4a, but also the thing that it's notoriously known for is its ability to kind of assist with neutrophil chemotaxis, and that's particularly high yield. So neutrophil chemotaxis chemotaxis with C5a, very similar to LTB4, right? We kind of had this discussion, interleukin 8. Those are your big players in terms of stimulating neutrophil chemotaxis to essentially take the neutrophil to the site of inflammation, taxing it over there using chemokines. And again, like we said, the terminal component of this complement cascade is going to be the C5b9 membrane attack complex. And this membrane attack complex, if you can imagine, you have some bacterial cell here. Okay, let's just say we have some bacterial cell. And essentially this membrane attack complex, what it does is it kind of just punches holes here in the membrane of the cell. So it's essentially going to be punching holes in the membrane of the cell. And so this cell will lose whatever gradient it has intracellularly to the extracellular space, and this can rapidly destroy pathogens. And this is essentially what this membrane attack complex is doing. Okay, it's essentially punching holes or perforating the cell membrane or capsid potentially of different pathogens, bacteria, viruses, etc. And where this is particularly useful in general is going to be against gram-negative bacteria. Okay, and this has to do with the cell structure, the cell wall, which is going to be covered more in the microbiology section. But in general, gram-negative bacteria are destroyed much more readily with the membrane attack complex, particularly Neisseria species in general. Okay, and this is going to come into play when we talk about terminal complement deficiencies. For example, if you don't have or can't form this membrane attack complex, you're going to have a really hard time controlling gram-negative infections, or particularly from Neisseria infections in general. So we'll touch on that in the next video. And the last thing that we kind of glossed over a little bit is inhibition with complement, right? So we talked about you can activate this complement cascade to go from C1 to eventually a membrane attack complex and a C3b component that can perform opsonization, but you can also inhibit different steps of the complement cascade. And I'm going to go into a lot more detail on this to come, but we have a delay accelerating factor, DAF CD55, and there's also a CD59, which I'll talk about coming up. And we'll go over the functions of these in more detail, but just to kind of introduce it here, we have activators and we have inhibitors that are going to affect different steps of the complement cascade. So in general, complement pathways always tend to be a topic that's very overwhelming in the sense that there's a lot of different steps. And we're not going to go through, every detail and every cofactor, but it is important to just have kind of a general understanding of this pathway and then, what factors we should be familiar with. So I'm going to go into a little bit of the detail and then we'll kind of zoom out and look at this big picture to try and understand, what do we really want to take away from this? And, and there are situations where you might get questions that are more detailed, but in general, you don't have to know every single detail. You just have to have an idea as to what's going on. Okay. And so that's essentially what we're going to try to kind of include here. And the thing that I want you to keep in mind as we're going through this, what are the things that you really have to know? Again, we really have to know these functional components of complement. Okay, that's the big thing. So if you had to take one thing away from all of this and you're watching this and you're like, I'm not really sure what I want to know. Number one is I want to know the functional components of complement and what they each do. Okay, so C3A, C4A, we talked about these being anaphylotoxins involved in anaphylaxis. When I say anaphylaxis, we're referring to histamine release from mast cells, right? C5A also does that, but it also stimulates neutrophil chemotaxis, which is particularly high yield. The neutrophil chemotaxis point, remember, we're getting neutrophils to the site of inflammation. C3b, very much involved with opsonization. We talked about that. And the other thing that we talked about was IgG, right? IgG also very good at opsonization. Okay, and then C5b to C9, right? We talked about membrane attack complex, basically perforating holes into the cell membrane. So those are all the big things to keep in mind, and you'll see them all come into play as we're going through these pathways. All right, let's start by talking about the classical pathway. Okay, so we're just going to kind of go through this one. Now, if you understand this pathway, the other pathways are actually pretty straightforward. Okay, so this is going to be kind of the steepest learning curve, but once we get through this one, the other ones will make a lot of sense, I think. So the first step, we're going to have to bind to our microbe. Okay, so this is our microbe. It's our pathogen. It could be a bacteria, it could be a virus, whatever. And on this microbe, you can see there's some IgG antibody. Now remember, IgG antibodies at the Fc portion, like we talked about in the previous videos, particularly that Fc portion that's closer to the hinge region on the antibody can bind complement. And what is it binding specifically? It's binding specifically C1. Now remember, it's not just IgG that can bind complement. It could be IgG or it could be IgM. IgM having more binding sites in the pentameric form can make it more likely for it to bind complement. So just another thing to kind of keep in mind when you're talking about, the structure of some of these immunoglobulins. But again, the point here, IgG is on the surface. It's tagging this microbe, right? So here it's binding to the epitope. We have our FC region portion down here, complement's binding to that, right near the hinge region. And we can see C1 is the component that's binding to it. Okay, so that's this C1 complex. Now this C1 complex, what it does, and remember what I said, we start at 1 and we end at 9, right? So it doesn't mean that we go necessarily in the correct order as we're going through the steps, but we do start at 1 and we end at 9, so that's something to keep in mind. So C1 complex is going to split C4 and C2. Okay, so you can see C4 here, you can see C2 here. It's essentially splitting them. And so one thing that I want to make kind of clear here with complement, if we start with something, let's just say we started with C4, and if we split it, when I say we split it, what am I talking about? I'm talking about splitting into 2 components, okay? So you're gonna get a C4A, okay? So you're gonna get a C4A, and then you're also gonna get a C4B, okay? So you're gonna get a C4A, and you're gonna get a C4B. You can think of this kind of subletter as being more of an activated form, whereas more of the inactivated form you can think of as just kind of having the number. Okay, so this being more inactivated and these being my activated forms. Now we already know what C4A does, right? C4A, we already said, stimulates histamine release, right? It's one of the anaphylotoxins. C4B though, what does that do? C4B is gonna go down into the next step. And the same thing is true for C2B. Okay, so both of these, all of the B's are gonna stay in the complement cascade. Now in the past, there was different notation for this, where the C2A was the component that stayed in the complement cascade, but kind of the updated way this is categorized is if it's going to be one of the B components, so if it's a C4B, if it's a C2B, if it's a C3B, that component can stay in the complement cascade, and that's the way to remember it. So if you get the B component, right, if it has the B here, it's probably, in some ways, possible that it's going to stay in the complement cascade. So if you have a B component, right, it's most likely going to be in the complement cascade here. Now C3B has some additional effects like opsonization, okay? But in general, these B components can stay in the complement cascade. This microbe marked with an antibody, the C1 complement factor bound to the FC portion on the IgG antibody, or it could have been IgM, and then that cleaved the C4 and C2 complement factors to C4b and C2b. Those come together and now they form a C3 convertase. So the convertase is telling you what this structure does. It takes C3, right? It would take a C3, just like we did with our C4, and it's going to cleave it into 2 components. Again, this C3 convertase is made up of C4b and C2b, okay, on the surface of this microbe. And you can see that happening here. So now we're going to, once we form this C3 convertase, okay, so C4b2b forms a C3 convertase. Once we form this, now a C3 can be coming by and we cleave that C3 into 2 components, just like we talked about. So we have a C3a. Okay, so C3a, we already know what that does, right? We set up here, functional components. C3a is also going to be involved with histamine release from mast cells. And the B component, we just said, the B component sticks around. Okay, so the B component comes down into the next step and helps form the C5 convertase. Okay, now the C5 convertase, it's like it says, once again, the C5 convertase is going to convert C5. Okay, and again, this is formed now from C4b, C2b, and C3b. Okay, so this is kind of where it gets confusing, but again, it's telling you what it does, right? So it's going to cleave C5. Now, once we finally get to this step where we form this C5 convertase, we're able to cleave C5, and again, we get our 2 components. We get C5a, which we already know what this does, right? We go up to our functional components here, right? C5a, we know that it's going to stimulate histamine release from mast cells. It's also going to be involved with neutrophil chemotaxis, right? So that's what C5a is going to be doing. And then we know C5b, it has a B in its name, so it's going to kind of continue this cascade. And we know we're home free because if we remember, the membrane attack complex, what is that made up of? Right, we just talked about this. It's made up of C5b to C9, okay? C5b to C9. And so this C5b component is going to combine with C6 through C9, right? So they're all going to come together and they're going to form the membrane attack complex, okay? So once we get the membrane attack complex, remember that's the terminal step of this complement cascade. Okay, there's really 2 kind of terminal steps that I think about. The first terminal step, like we talked about in our first video on this, was going to be C3b. C3b, it has a B in the name, right? We said if it has a B in the name, it can continue down this cascade. So C3b can continue to form the C5 convertase, right, in the next step, or the C3b can then go on to stimulate opsonization. So the C3b can do either one, right? That's kind of the first terminal point of this cascade is opsonization from C3b formation. But the second terminal point of this cascade is going to be the membrane attack complex, in the sense that this is a structure that isn't going to go on to another step, and it's a structure that can punch holes into this microbe's membrane. Then we're getting into the common pathway, right? And that essentially we're saying common because all of these different pathways, right, they're going to have some slight differences up here, but once you get to this C5b step, you can see they all have C5b here, you're essentially going to be going down the same pathway, right? You get the C5b, the C5b is going to bind to C6 through C9 and form the MAC. Okay, let's go through the lectin pathway here really quickly. So the lectin pathway, and the nice thing about this lectin pathway is it's essentially the same thing as the classic pathway. The only difference is in the first step. In the first step of the classical pathway, we had IgG or IgM antibodies bound to the microbe surface, and so we bound our C1 complement to that. But in this situation, instead, you're going to have mannose, right? You're going to use these mannose structures on the microbe surface. And you're going to have mannose binding lectin, right? Mannose binding lectin, MBL, is going to bind to the mannose, okay? And then that structure is going to kind of act as that initial C1 from the classical pathway. Every step after is going to be the same. This mannose binding lectin, just like C1, is going to cleave C4 and C2. The B's are going to stay. We know what the A's do up here. The B's are going to stay, so the B's are going to come together, and they're going to form a C3 convertase, which tells you what it does. It converts the two components of C3 to C3A, which again, we know what that does, right? It's one of our functional components. And then C3B can either go on to do opsonization or can go into this next step to form the C5 convertase. C5 convertase, it says what it does in the name. It converts C5, right, to C5b and C5a. C5b can combine with C6 and C9 to form the membrane attack complex. Go right back down the common pathway. So the only difference between the classical pathway and the lectin pathway is the first step. Okay, so that's where, if you're kind of thinking, okay, well, how would they ask me a question? Do I have to know all the details? You want to know the differences, right? So you want to know that the classical pathway is associated with immunoglobulins, and the lectin pathway is associated with mannose binding lectin, kind of triggering the cascade. The alternative pathway has to do with C3 starting things off. Okay, so it's a little bit trickier. So let's just go through that really quickly. So the first step has to do with C3 essentially binding to the cell surface to form C3b. Now, once you get C3b here on the cell surface, okay, which again is completely different from what we talked about with the other two pathways, there's also going to be a second component, a factor B. And when that factor B gets cleaved here, you end up forming a C3 convertase. Now, like I said, I'm not going through every single detail here because it's just not high yield, right? We don't need to know every single cofactor. But I am going into some detail because, this is one of those things where if you understand this a little bit, you can make a lot of sense of the disorders and see where some of this stuff comes from. So with the alternative pathway, it's a little bit harder because it doesn't have the same features as the other two pathways initially. Your C3 convertase is actually different. Okay, so if there was a question where they wanted you to distinguish what makes up a C3 convertase, right, you can see in the alternative pathway, it is completely different than in the classical and the lectin pathway. Okay, so this is made up of C3b and then this B factor sub B component. Okay, and then this eventually what it does is it acts just like the name says though. A C3 convertase is cleaving C3, and so you're going to get further C3 cleaved. You're going to make more C3b, right? That can go back into this pathway, just like in the previous steps, and form a C5 convertase. So again, after those first initial steps, everything, beyond the C3 convertase is the same, once again. So if you understand everything beyond the C3 convertase pretty much, you can understand each of these pathways. It's just that the first step is a little different. So let's just go through the text here just to hammer this home. So again, when we're talking about the classical pathway, we're talking about immunoglobulins setting things off. Which immunoglobulin specifically? IgM or IgG? You get a C1 complex that can bind to the Fc portion, splitting it C4 and C2. When we split something, we get the A and B components, right? We know where a lot of these structures go. If it has a B in its name, though, it can stick around. Okay, so if it has a B in its name, it can stick around. So the C4b and 2b stick around. They form the C3 convertase. We cleave C3. We get the B component that sticks around. Now we form a C5 convertase, right? And that takes us right into the common pathway, where we'll then form C5a, C5b. The B sticks around, helps form the membrane attack complex. Okay, lectin pathway, same thing. The only difference is we activate with mannose
and we have mannose-binding lectin that forms the C1-like complex, because it does essentially what the C1 complex does. Everything else is the same as the classical pathway. Alternative pathway, the difference is we're activating on the microbial cell surface with C3b bound to the surface. It's going to bind this factor B, and then we're going to form a C3 convertase, eventually forming a C5 convertase once we cleave C3b and add that to our structure. Okay, and so again, we get to the common pathway. It's the same thing. They're all going to end in this membrane attack complex formation. Okay, so they're all going to eventually get to that step. They just start a little bit differently. Okay, so the C1 esterase inhibitor, and we'll talk about the relevancy of this with C1 esterase inhibitor deficiency, but the C1 esterase inhibitor is going to prevent this first step. So if I had some, let's just put C1 C1 inhibitor here, it would essentially inhibit what this C1 component is doing. And what is it doing? It's cleaving C4 and C2, right? So it's inhibiting this step. So you can imagine if you had too little of this, if you had a deficiency of this C1 esterase inhibitor, you wouldn't be able to stop C4 getting converted to C4b, C4a. You wouldn't be able to stop C2 being converted to C2a, C2b, right? So you would have a lot of complement activation in this initial step. You'd have very low levels of C4 because you wouldn't be able to inhibit or regulate it going into form C4b. Okay, so that's why low C4 levels is very classic for C1 esterase inhibitor deficiency for that reason, right? Because you can't inhibit this step. So this C4 is constantly getting converted, right? Not regulated. All right, the next one on here, the next inhibitor is going to be delay accelerating factor, also known as CD55. And we see a deficiency of this with PNH, paroxysmal nocturnal hemoglobinuria, which we'll talk about in a minute. And this essentially prevents the formation of the C3 convertase. Okay, so you can see, where's my C3 convertase? I have them here, right? So decay accelerating factor is going to inhibit this step, right? It's going to inhibit these two structures coming together to form the C3 convertase. In our body, right, in homeostasis, these molecules like decay accelerating factor, what they're doing is they're regulating things, right? So if you have too much decay accelerating factor, you're not going to be able to have complement mediated destruction. But if you have too little of it, you're going to have too much destruction, right? So that's the concept. That essentially these are helping to regulate this process so we don't have activation. And the final one on here is the membrane inhibitor of reactive lysis, CD59, which is going to prevent the formation of the membrane attack complex. So in all of these, right, it's going to combine with C6 through C9, and this is going to make our MAC, right, which is essentially going to be C5b through C9. And what this CD59 is going to do is it's essentially going to inhibit at this step. Okay, I'll just put MIRL here for membrane inhibitor of reactive lysis. Okay, so that's what that stands for. And again, it kind of looks like the MAC name, and that's maybe one way to remember it. And it's essentially inhibiting this step, preventing the formation of the membrane attack complex. All right, so let's put together kind of everything that we've been talking about to go over some of the disorders of complement. Probably the least high yield of the four on here is going to be this early complement deficiency. It's just like the name says, it's early, right? So it's in the first few stages. So you have some deficiency. It could be C1, C2, C3, C4. It's pretty nonspecific. But in general, if you had a deficiency of these early complement factors, the patients are more likely to have respiratory and sinus infections in particular. Okay, and there's an increased risk of developing lupus. And now remember, lupus, we tend to see low levels of complement, particularly the early complement in the setting of lupus. And you can kind of make a case, well, is that because you have more complement activation? Maybe you have an autoimmune hemolytic anemia, thrombocytopenia, etc. Is it kind of the chicken or the egg, which came first? But in general, there is an association here, so just keep that in mind. Like I said, probably the least high yield of the four, but it's good to have it in here just for inclusion's sake. What I would focus on that's much more high yield is the terminal complement deficiency. And the biggest thing that you have to take away from this is that these patients are at increased risk of Neisseria infections. And there's some repercussions of this where this can turn into kind of a more complicated question stem that I'll talk about in a second. But in general, what you really want to know, what you really want to take away from this is if for whatever reason there is a deficiency of C5 to C9, right? Particularly, we're talking about C5b to C9, right? That's going to form that membrane attack complex that we talked about. And that's going to be very much associated with, like we said, perforating pathogen membranes, particularly effective, like we said, against gram-negative bacteria. And the big one that we always talk about, again, being Neisseria. Okay, and we'll talk about this in a little bit more detail in just a minute. Before we do that, though, let's jump into talking about the C1 esterase inhibitor deficiency. So again, the general concept here is, right, if I have some microbe, right, this is going to be my pathogen, right? And then I tag this thing with an antibody. And then remember my FC portion here, if it's IgG or if it's IgM, let's just say it's IgG, this FC portion, particularly close to the hinge region, I can have complement formation. So I can have a C1 complex come here, and then I can take C4 and C2 and essentially convert these to C4A, C4B, and C2A, C2B. And I know the B's are going to stay, and these can come together to form my C3 convertase, which is going to be used to take C3 and then cleave C3, right, to C3A and C3B. But the point here is that for, if I have C1 esterase inhibitor around, that can inhibit this C1 from acting, right? It's just like the name says, C1 inhibitor. Okay, so it can inhibit the C4 and C2 from going down this pathway, and this is used to regulate, overactivation of complement and that type of thing. Now, in some people, though, they have a deficiency of this C1 esterase inhibitor, and the presentation they have, one of the things you'll see is that their C4 levels are very low. And again, you can make sense of this, right? So if for whatever reason you didn't have this C1 esterase inhibitor, let's do this in another color. Let's just say you didn't have the C1 esterase inhibitor, okay? So you didn't have it, or you had really low levels of it. What would happen is it wouldn't be able to inhibit the C1 component, right? And so if it wouldn't be able to inhibit the C1 component, this C1 component might go kind of overactive, right? Because it's not going to get regulated at all. So you're going to have more and more of this C4 and C2 getting converted to their subcomponent, C4A, C4B, right? So what's going to happen to your C4 levels in the blood? They're going to go down, right? Same thing with the C2 levels. So you might have very low C4 levels, and that's typically one of the tests that come up when they're asking questions about C1 esterase inhibitor deficiency in general. There's a lot of stuff written here about bradykinin, so let's talk about that. Now, we said earlier that bradykinin in general, when we were talking earlier in this playlist, we said that this is very much involved with things like vasodilation, right? Vasodilation. And we also said it was very much involved with the pain response. When we feel pain, that has to do with those high levels of bradykinin when we have inflammation. And, bradykinin comes from this high molecular weight kininogen. Okay, I'm just going to write it HMWK, sometimes written as kininogen, but we'll just write it as high molecular weight kininogen, can form bradykinin. And what causes high molecular weight kininogen to form bradykinin? That would be kallikrein. So kallikrein will help stimulate the conversion of high molecular weight kininogen to bradykinin, and then bradykinin causing vasodilation and pain. So you can imagine if you have an inflammatory response, you're going to have higher levels of kallikrein, so you get more bradykinin around. That's the concept. We need regulators, right? We need some kind of regulator of this process. And so the regulator here is your C1 inhibitor, C1 esterase inhibitor. Actually comes in and it says not only does it inhibit C1 in the complement cascade, but it also inhibits kallikrein, again, keeping things in check, right? So things don't get out of control, and that's what the C1 esterase inhibitor is doing. Now, again, going back to the point I just had, if for whatever reason a patient had a deficiency of the C1 esterase inhibitor, now it can't inhibit kallikrein. So what happens? Well, now kallikrein can go unregulated, right? It can go unregulated, and it can just stimulate this process to a very high degree. And so what are you going to get? You're going to get very high levels of bradykinin. You're going to get a lot of vasodilation, right? And this can also cause vascular permeability to increase, and you can end up with edema. Okay, so you can end up with increases in vascular permeability, and you can end up with edema. And these patients can also have a lot of pain because of these really high levels of bradykinin in the setting of a C1 esterase inhibitor deficiency. So these patients typically, this will be autosomal dominant. Okay, remember, we always try and remember the autosomal dominant diseases, the X-linked recessive and X-linked dominants, and then anything that doesn't fall in those categories, that's going to be autosomal recessive. Okay, that's kind of my way of organizing it. And again, we have increased levels of kallikrein. We already talked about that. The kallikrein is going to basically go unregulated, and that's going to lead to increases in bradykinin. Now, the presentation typically, and there's more detail on this, we're just going over the high yield stuff here. Typically, these patients will have recurring angioedema. They might have facial edema. They might have abdominal edema where they have abdominal pain and nausea from edema that's happening there, or even edema in the extremities. Okay, the concern here is we don't want these patients to get laryngeal edema, where we start to close off the airway. Okay, so this is going to kind of present almost looking like this patient is, potentially going into anaphylaxis, or this patient had some allergen exposure. But one thing that's different when you're talking about C1 esterase inhibitor deficiency from something like a type 1 hypersensitivity is that in this situation, there's really no urticaria. Okay, especially in a board question.
There's no urticaria. So that's one way that you can try to tease out some type of type 1 hypersensitivity leading to anaphylaxis versus a C1 esterase inhibitor deficiency, because they're treated differently, right? Because if I have a patient that has anaphylaxis, I might be thinking epinephrine. If I have a patient that has C1 esterase inhibitor deficiency, I can try and get these patients C1 esterase inhibitor because that's what they're missing. That's going to slow this pathway down. If I give them concentrated C1 inhibitor, that will inhibit this kallikrein and we can get the bradykinin levels under control. So they're treated differently. So it's important to distinguish them. One thing they'll say is that the patient recently received an ACE inhibitor or was started on an ACE inhibitor. So we talked a little bit in the cardiology section about this, about how the ACE enzyme, it has a few different functions in the RAS cascade. But one thing that it does is it actually inhibits bradykinin, okay? So if we had a patient that had an underlying C1 esterase inhibitor deficiency and we started them on an ACE inhibitor recently, okay, so if we just started them on an ACE inhibitor and they already have low levels of C1 esterase inhibitor, look what's going to happen. If I put somebody on an ACE inhibitor, I'm going to inhibit this enzyme, right? And so now if I inhibit this enzyme, I'm not able to regulate my bradykinin. So not only will my kallikrein be, stimulating bradykinin, so there's more and more bradykinin being produced potentially, but I can't even inhibit the bradykinin with the ACE enzyme to try and keep things under control. So it's kind of like double trouble here because I have an ACE inhibitor that's inhibiting one thing that's regulating bradykinin, and then I don't have my inhibitor to control kallikrein, so I have even more bradykinin. And so that's typically how this will come up. So these patients should avoid ACE inhibitors, but a lot of times in a board question, that's how it's presented. They were started on an ACE inhibitor for high blood pressure or whatever, and now they're presenting a few days later, potentially with new facial edema or abdominal pain, recurrent abdominal pain with new edema, etc. Okay, so that's usually going to be the trigger. The trigger can also be trauma, stress, right? There are a lot of different things, but that's one classic way that this comes up. All right, PNH, paroxysmal nocturnal hemoglobinuria. So this is classically going to be a hemolytic anemia. You can have effects on some of the other cell lines, but essentially what's happening here, if I just draw in a red blood cell here, I'm going to have certain proteins on this red blood cell. We're going to call them GPI. And so these are going to be membrane-bound glycerol, phosphatidylinositol proteins. Okay, so GPI proteins. And these proteins are going to be things like CD55, right? We already talked about what CD55 does. This is your decay accelerating factor. It's going to inhibit the C3 convertase that we talked about when we went through the complement cascade in the last video. Okay, you're also going to have other GPI proteins on here, like CD59, which is going to inhibit the membrane attack complex, right? And that's going to be the membrane inhibitor of reactive lysis. Okay, so we already talked about those. So these are going to be on the cell surface of my red blood cell. Okay, so this is my red blood cell. So in a way, I kind of want these things on my red blood cell because I don't want my red blood cell to get destroyed by complement that's floating by. I don't want complement to get activated on my red blood cells. Now, if for whatever reason, though, I didn't have these proteins, okay, let's just say I didn't have these proteins on the surface of my cell. Well, if I didn't have them here, it might be more likely that I potentially could have complement activated on my red blood cell. And that complement cascade that eventually leads to a membrane attack complex forming, right, would destroy my red blood cell. And that's essentially what PNH is. So it's complement destruction of red blood cells due to loss of these GPI proteins. Specifically, CD55, right, we talked about that. That's a decay accelerating factor, and CD59. Okay, so those are the two that you really want to associate here with this paroxysmal nocturnal hemoglobinuria. We're inhibiting the inhibitor again, or we don't have the inhibitor, so to speak. So we have overactivation on our own cells. It doesn't just have to be on red blood cells. It could be on white blood cells. It could be on platelets. You could have a pancytopenia with this potentially. So again, in general, and you'll see this a lot when you're talking about hemolytic anemias, you see this in lupus, these patients will be very fatigued, right? They're going to have much lower oxygen carrying capacity in general. Systemically, they're going to have pallor, intermittent jaundice. Where's the jaundice coming from? We're going to have all of these red blood cells, right? We're going to have bilirubin that's being released, that's causing some of this jaundice. And then we'll have some hemoglobinuria, potentially with kidney injury, because of all of the red blood cell destruction. Now, the very classic thing, though, that you'll sometimes see with PNH is going to be this venous thrombus in atypical locations. So the idea here is as you're destroying these red blood cells, like we just said, you're going to have breakdown that eventually leads to high levels of bilirubin in the blood, but you're also going to have hemoglobin. And hemoglobin can cause oxidative stresses onto the endothelial walls. So this hemoglobin can exert oxidative stresses,
right? Oxidative stresses, oxidative radicals can start to damage some of the walls here, particularly in areas where there's a little bit more venous stasis, okay, where there's blood not necessarily moving as fast, and that's going to be more on the venous side. And this can eventually lead to clots forming, right? We can have venous thrombus that form. And again, the classic thing is you typically aren't going to see venous thrombus in the hepatic veins, for example, right? The hepatic veins, renal veins, or the cerebral veins, right? So you're going to start to see kind of these very unique locations, not the typical, deep venous thrombus in the leg, right? These are going to be atypical locations in board questions as kind of a call out. So if you see patients that have intermittent jaundice, pallor, fatigue, hemoglobinuria, particularly hemoglobinuria that's worse in the morning, very classic for PNH, okay? And why I say worse in the morning is because if a patient gets up to have their first void in the morning, the urine's typically going to be more concentrated because they were sleeping and they weren't consuming fluids. So a more concentrated urine plus the fact that you're spilling hemoglobin, that's going to tend to be the darkest urine, where you're going to have very dark red appearing urine, okay? Very classic for PNH, dark urine in the morning. When you have this venous thrombus in atypical locations, particularly in the hepatic veins, that's sometimes referred to as Budd-Chiari syndrome. We'll go into way more detail on this when we get to the GI section, but when you start to block off the blood leaving the liver, right? All that blood is going to start to sit in the liver. You're going to end up with this congested liver that sometimes starts to form what's called a nutmeg liver, okay? And we'll get into more detail with that, but eventually that leads to a liver failure. And you're going to get a backup of a lot of that venous blood. You're going to get things like ascites, peripheral edema. One thing you won't get usually is going to be JVD, because the backup on the venous side is proximal to the jugular veins, right? If you had JVD, you'd be thinking more about something like CHF, okay, which we went through a whole bunch of detail on this in the cardiology section too, but you just have to think about this anatomically speaking, right? The hepatic veins are going to drain eventually into the inferior vena cava, and the blockage, if you had a hepatic vein thrombus or Budd-Chiari syndrome, would be before the hepatic veins reach the inferior vena cava. So you wouldn't expect, for all those reasons, jugular venous distension, okay? But you would expect things like ascites, peripheral edema, for that reason. Okay, now if you're going to diagnose this, the Coombs test is negative. And I don't want to get too off track and go down this rabbit hole just yet, but we will talk in great detail about the Coombs test. This has more to do with antibodies on the surface of these cells. Again, the point here is not that we have antibodies that are destroying the cells. The point here is that we're missing our own inhibitory, regulators of complement on our cells. All right, and the last thing here is there are certain drugs that you do want to be familiar with. Now, step one isn't as big on testing medications, but this drug in particular, the eculizumab, is an anti-C5 antibody. Okay, and so the idea here is if I'm destroying my own cells, right? If I'm destroying my own cells because I don't have my CD59 and my CD55 to regulate things, to inhibit complement from attacking my cells, I can try to attack the complement that's attacking my cells, okay, as crazy as that sounds. So if I had an anti-C5 antibody, okay, so here's some antibody, and let's just say, right, that I have, let's go back to my red, let's clean this up and go back to this red blood cell here. So here's my red blood cell again. It doesn't have the CD55 or 59 on it, and let's just say that for whatever reason, right, complement starts to get activated here. And again, it's probably going to start with C1 complex or C1-like complex. That's eventually going to lead to the formation of a C3 convertase. And eventually, it might have some C5 that binds, right? When I get to my C5 convertase. What these anti-C5 antibodies do is as this is happening, right, as this is kind of going through the complement cascade, these antibodies can then bind to that C5, right? So here's some C5. It can bind to this and destroy it so that we never really get to a point where I form C5b and then form my membrane attack complex. I can't get to that next step to actually destroy the red blood cell, all right? And that's what this drug is doing. It's a monoclonal antibody, right, that ends in mob, eculizumab. And there's also another one on here, not as high yield. But the point is if I use this medication, right, which may work very well, the issue is that if I can't form membrane attack complexes in this patient anymore because I'm inhibiting C5, what would that predispose this patient to? That would predispose them to Neisseria infections, because I essentially am giving this patient a terminal complement deficiency so I don't destroy their red blood cells, right? That's essentially what we're doing. And so because of that, now these patients are at increased risks of Neisseria infections. So if you give a patient eculizumab, you have to know that this patient is going to be at increased risk. So you're going to have to make sure they're vaccinated from Neisseria. And in some cases, they might even be on prophylactic antibiotics to prevent certain infections, particularly, again, Neisseria being the big one. And that's something where you have to, that would be a great board question because you're integrating so many components of your knowledge here. So that's the way you have to kind of think at some of these multi-level questions, especially when you go on to step 2, when you have to have a little bit more wider of a knowledge base in terms of what's going on.
Take 15 seconds to pause the video, read the question, and then we will go through the answer. All right, so we have a 37-year-old female who's presenting to the emergency department with a 3-day history of worsening severe left lower quadrant abdominal pain. As you get more kind of into the clinical realm of things, right when you see this first sentence, you want to start kind of jogging a differential in your mind, right? So when you see severe left lower quadrant abdominal pain, again, how old is she? She's 37 years old. Of course, you want to start to think a little bit about things. I mean, for me, ectopic pregnancy is definitely up here. What other things would you be thinking about potentially? You could be thinking about a UTI maybe pyelonephritis potentially, if she has a fever and they say she has CVA tenderness or that sort of thing. Then also diverticulitis would be pretty atypical. Usually in a board question, if they're asking about diverticulitis, there usually is left lower quadrant abdominal pain, but it's usually in an older patient typically. But again, there could be a variety of things. Kidney stone would be another one. We could just put nephrolithiasis. So, as you kind of get more comfortable with these clinical vignettes and, especially when you get into the clinical world, right when you hear a sentence like this, immediately you kind of have a set of differentials that you're starting with, right? And so this is kind of one way to think about it. All right, so let's keep going here. So the patient states that she ordered a hamburger from a fast food restaurant for dinner the day prior to onset of symptoms. Okay, so that might be relevant potentially. E. coli potentially as like an infectious cause. The patient's past medical history includes allergies to dust, pollen, and numerous medications. Okay, so they're kind of giving us a lot of information from some different areas here. Additionally, the patient was started on lisinopril by her primary care physician 1 week prior for essential hypertension. Okay, vital signs are stable. Physical exam reveals significant edema in the bilateral upper extremities. Okay, that is interesting. Let's keep reading here just to get a little bit more information. When questioned regarding the swelling, the patient appears unconcerned and states, "Everyone in my family has this happen from time to time." So this is definitely a big piece of information here. A lot of the time when they put what the patient says in quotes or anything like that, especially for clinical vignettes, board questions and that sort of thing, it tends to be relevant. So we'll come back to this in a second. We'll finish here. Abdominal CT scan reveals significant diffuse bowel wall edema. Okay, so this would explain a little bit about where this abdominal pain is coming from. Which of the following sets of laboratory data are most consistent with this patient's presentation? Just putting the pieces together here. So this patient was recently started on lisinopril for essential hypertension about 1 week ago. Okay, so lisinopril, so this is where you have to, put the pieces together. You have to understand lisinopril is an ACE inhibitor, right? So it's an ACE inhibitor. And the other key piece of information here is that this patient seems to have a family history of frequent swelling or edema, right? When questioned regarding her swelling, she says she's probably unconcerned about it and everyone in her family has this happen. So that would suggest potentially that there's some family history of swelling or edema. So just kind of putting these pieces together, we're probably dealing with a patient that has hereditary angioedema. A lot of times in board questions, they'll present this more with like lip swelling or facial swelling, but it can happen in the extremities. It can happen in the bowel too. And in this case, this is probably what's contributing to her severe left lower quadrant abdominal pain. So she was already kind of predisposed to this angioedema, and then you gave the patient lisinopril, and that kind of tipped her over the edge. So let's just kind of review that process really quickly. So just remember the key player here is going to be bradykinin, right? So bradykinin, and this is exactly what they're asking about here. They're really asking about this pathway. And this is why I tell you for step 1, you got to understand the pathways. If you know the pathways, you can kind of rule in and out different things. You just have to make sure that you're solid on your mechanisms and your pathways. Okay, bradykinin is formed from high molecular weight kininogen. And then this bradykinin, just remember the functions of bradykinin, right? So some of the functions, one big one is vasodilation. So this can lead to edema, interstitial edema, right? As we start to vasodilate and we increase the permeability of capillaries and that sort of thing, that can definitely lead to edema. And with this vasodilation, like we said, there's increased, I'll just put perm here for permeability, right? This is all contributing to the edema. And sometimes we can also get pain,
right, from bradykinin. So these patients can complain of painful edema, or in this case, this patient has abdominal pain, likely secondary to the elevations in bradykinin and this diffuse bowel wall edema. What is really regulating the step here is kallikrein. It's kind of stimulating the conversion of high molecular weight kininogen to bradykinin. Notice on here, when we're looking at this lab data, they also included, the C2 level, the C4 level, C1 inhibitor functions. Let's just talk about that too, because this is all kind of interrelated here. So also remember, and we talked about this a little bit in the video lessons if you've been following along, but remember that C2 and C4 can get converted to their subcomponents here, C2b, C2a, and C4a, and C4b. And this all happens by C1, right? C1 is going to allow for a lot of these processes to happen. Now, remember that there's a C1 inhibitor that regulates this process. So I'm just going to kind of draw this here. There's a C1 inhibitor that can regulate this process. So essentially, to put this all together, what happens here in this hereditary angioedema is that we have some dysfunction in the C1 inhibitor. Okay, so the C1 inhibitor has some kind of dysfunction. Now, there's different types of hereditary angioedema. A lot of this is beyond the scope of step one. In some cases, you can have low C1 inhibitor. I'll just put an I for inhibitor. But in all the cases, you have dysfunction of the C1 inhibitor. And so that's the thing that everyone should remember, right? So the C1 inhibitor is not going to be functional. It could be low if it's a certain form of hereditary angioedema. But in general, whether it's low or whether it's normal values, in both forms, the C1 inhibitor has some form of dysfunction. Now, you can imagine if the C1 inhibitor had some dysfunction, right? So if I knocked out the C1 inhibitor here, what's going to happen? Well, now this complement, the C1, is going to kind of be over stimulated, right? Because there's nothing to regulate it. There's nothing to keep it in check. And so you're going to get a lot of C2 and a lot of C4 getting converted because there's nothing inhibiting this C1, right? And so what happens is the levels of C2 are going to drop and the levels of C4 are going to drop because they're getting converted pretty rapidly here. And there's nothing to inhibit this process because we have a dysfunction or deficiency of this C1 inhibitor. That's the point. So with that being said, we know from this fact that we should have low levels of C2 and C4. Right? So if we're just kind of scanning this thing here, there's really only two options where we have low C2 or C4 levels, which actually is kind of nice. It simplifies this quite a bit. You can see that there is an option here where one is low and one is high, but again, we would expect both of them to be low in this situation, right? If we just kind of understand the mechanism. The other thing here is you can actually solve this problem just by knowing this and knowing that, hey, the C1 inhibitor function is going to be impaired, right? C1 inhibitor function is going to be impaired. We pretty much know that F is the right answer, right? Just based off of that information, we can solve this problem. But let's just explain why the kallikrein and the bradykinin are also elevated really quickly, okay? Because you may be in a situation in the board question where it's not quite this simple, right? You might have to literally go through every single arrow. So kallikrein and bradykinin are elevated. Why is that? Remember that the C1 inhibitor can also inhibit kallikrein, right? So if it inhibits kallikrein here, what's going to happen? So if kallikrein is inhibited, that's going to regulate this process, this conversion of high molecular weight kininogen to bradykinin. If I have a patient that has hereditary angioedema and they don't have C1 inhibitor or they have a deficiency or dysfunction of the C1 inhibitor, then what happens? Well, if we don't have that C1 inhibitor functioning correctly, right, the kallikrein is going to kind of be overregulated, just like we saw down here. So the kallikrein is going to be more stimulated, right, because there's nothing to kind of keep it in check. There's nothing to inhibit it. And so we're going to get more high molecular weight kininogen converted to bradykinin, which means that we're going to have higher levels of bradykinin, right? And with higher levels of bradykinin, we get more vasodilation, more vascular permeability, more pain, more edema, which is exactly what we see here. Also, the last kind of layer here is to remember that ACE inhibitors run into this pathway also. So remember, this patient was given lisinopril, which is an ACE inhibitor. So to integrate ACE inhibitors here, remember that the ACE enzyme normally inhibits bradykinin. Okay, so the ACE enzyme, again, as a regulatory step, inhibits bradykinin. When you start somebody on an ACE inhibitor, right, you're going to inhibit this ACE enzyme. Okay, so if I start somebody on an ACE inhibitor like lisinopril, what's going to happen is I'm going to inhibit that ACE enzyme. If I inhibit that ACE enzyme, that's only going to further potentiate or stimulate this bradykinin, right? I'm going to get more and more of this bradykinin or more functioning bradykinin because I'm now inhibiting this enzyme. This was kind of like the last checkpoint. This patient at baseline probably didn't have a C1 inhibitor, so they already had kind of high levels of bradykinin, but now we added an ACE inhibitor and we actually made that problem even worse. And so again, the key is you can sit in a textbook and you can memorize, oh yeah, hereditary angioedema, so it's associated with edema, and ACE inhibitors make it worse. But the hard part is seeing that in a board question and just saying, hey, this is an ACE inhibitor, this patient has bowel wall edema, right? They have a family history, boom, you're thinking hereditary angioedema. Now you just have to understand the mechanisms to apply that knowledge. And so for those reasons that we just mentioned, again, the kallikrein should be elevated, right? We just said that. And the bradykinin should also certainly be elevated. So F here would be the best answer.
Hey everybody, Dr. Vizo here. In this video, we're going to talk about vaccinations. We're going to build a little bit on some of the other